So the antibody purpose is to do one, make complement, just like you just said.
Complement cascade.
2, opsonization.
And that's what's mixed one.
Yeah, opsonization is when we like add the ketchup and the mustard to the hot dog, and we're basically then we can make the cells more tasty to the macrophages to eat out the bacteria. What else does it do?
It inactivates it, but I don't really remember what that means exactly. Like it just inactivates the.
Say you have a cell that got infected with the virus. So remember how I talked about viruses actually come attached to the cell and inject their stuff in it, and they basically hijack a cell, right? Basically, like they hijack the cell and they start joyriding, and they start using all this. They start using that cell to produce more virus proteins. And so if you inactivate that cell, you'll stop producing those proteins.
Okay.
Does that make sense?
So you pretty much kill it in a way.
Yeah. It's not really like destroying the cell; it's just stopping it from
producing what it needs.
Yeah,
to
it's like shutting down the factory. The factory is still there, but you shut the factory down.
Gotcha.
Which is producing viruses, so you want to shut it down.
Yeah.
Does that make sense? What else does it do?
Agranulation.
Agranulation, right? Which is what? What is agranulation?
It's clotting of the cells together so that the bacteria won't be able to like reach other cells.
Yeah, so it's kind of like I like to use the word clumping. It kind of clumps it together. So it's like if you have a bunch of bacterias and stuff like that around, then you have a certain antibody called what? IgM, right? So remember M has 1, 2, 3, 4, 5.
Okay.
That's right there.
So together it makes like a clot.
So it makes like that pentamer, right? Remember this thing?
Okay.
Remember that thing kind of looks like a snowflake on the board? He kind of had that picture of it.
Yeah.
And so that's the whole purpose is, well, he'll grab onto like the infected cell or bacteria or whatever it is, the antigen right there, and then he'll grab onto another one, and then he'll grab onto another antibody sequence.
So another snowflake.
Exactly. All the while he's grabbing onto another antigen or whatever it is here, then another pathogen, and then this guy's grabbing onto another one. All while he's grabbing onto some, does that make sense? And so these antibodies, these IgM antibodies actually come together, they group together, and they start grabbing each other's arms, all while they're starting to grab these pathogens. And when they grab those, then they kind of do this like granulation, which is when it starts to clump all these things together. It starts to make them come into one area.
Okay.
Makes them a lot easier to kill if they're all in one area. Does that kind of make sense?
Yeah.
So that's why I think of it as...
I feel like, because this is in your lymphatic system, so could that like stop the flow of...
It definitely would make things a little bit more thicker, and it would make things have those little bumps in it, like those little like granules, like an RH thing you did in the blood. Did you do that, lab?
Oh, yeah.
It kind of makes those little granules.
Yeah.
You definitely would make those. I mean, if you had tons, then yeah. And does that make sense why your lymph nodes swell then?
Why you would swell?
Your lymph nodes swell when there's an infection.
Because you have clumping in the area.
Yeah, but it's not only these, it's just you have a huge production, you have a huge mass influx of like B cells within the lymph nodes that are going on. So it's starting to grow, right? Because you have tons of B cells coming up, and they're causing a bunch of these things to happen.
Okay.
Right? Because complement will cause inflammation, right? And so if you're releasing a bunch of antibodies, and one of the things that antibodies do is release complement, and complement actually causes inflammation. Well, if you have inflammation, they're going to get swollen, right? Mm-hmm. And so you'll have swollen lymph nodes if there's an infection in that area of the body.
Okay.
Does that make sense?
Yeah.
Was there one more?
IgE and histamine release.
There we go. And that's with what kind of cells? Perfect. Basophils and mast cells. So mast cells and basophils actually have this pretty cool thing they do, right? They're full of this thing called histamine, and so they'll actually release histamine, right, in the presence of IgE.
Mm-hmm.
And so we have IgA, which is found where?
I have no idea.
So IgA is going to be mostly like the mucous membranes.
Oh, yeah.
Sorry. So it's
So it's gonna be like in your mouth.
yeah. So anyways, mucous membrane, you'll find a lot more IgA present. IgA antibodies present.
Okay.
And like, what's the most common antibody in your body? IgG.
IgG.
Yeah, IgG is the most common antibody in your body.
Most common.
It's the one that you have the most number of at any given time.
IgG has 2 G's, so it's most common.
And then you got IgE. His whole thing is IgE will actually, like, if this is the mast cell, let's say it's a mast or a basophil cell.
Uh-huh.
Well, the IgE looks like, oh, come back. IgE kind of looks like this, right? Just as a monomer, right? Because you got the different shapes. You got that one, and then you also got like this one, and the pentamer. Well, IgE will come down, and it'll stick its little butt inside there, okay?
Okay.
And when it does that, it activates the mast cell to start spraying a bunch of histamine. And when it sprays a bunch of histamine, the histamine then leads to vasodilation and inflammation, right?
Uh-huh. Wait, it does vasodilation and what else?
Inflammation.
Oh, okay.
They become inflamed. Sorry. I don't know why I'm yawning so much. But yeah, so it'll become inflamed. And inflamed just gets red and swollen, right?
Yeah.
And so you'll have bright red because of vasodilation, bringing blood to the surface. So it portrays that reddish color. And then it'll be swollen because of what?
Because the blood's, or the fluid's going to be coming in.
Coming where?
Into the tissue.
In the tissue.
Because
Why?
it's vasodilated, because there's so much.
Causes an increase in? There's the P.
Plasma?
Permeability. Perfect.
Wait, so we're vasodilating the,
Artery.
or it's going to be the lymphatic system, right? So
Arteries. Arteries, really. This causes arteries. This is called arterial dilation.
Okay.
So vasodilation is going to
in
be.
the arteries.
Yeah.
So we have more blood flow through those.
Mm-hmm.
But would that necessarily make it go out into the tissue?
Yeah. So do you remember when we talked about like, so like this is your normal artery.
Okay.
It's like this. Remember?
Yeah.
And so remember how I can kind of see through those little tiny cracks in my hands, just little tiny cracks there. And so if I scooped some water up and I held it, it'd hold the water pretty good, right?
Okay.
But it would leak a little bit. So it's a kind of semi-permeable. It's not very permeable though, right? The water can go through. And so it's kind of how it is with vasodilation. What happens is I'm sitting like this, and I just kind of stretch my vessel. I go, and I stretch it.
And then now it's more permeable.
So now you see those bigger holes, and so like you said, it's more permeable. And so vasodilation increases the permeability of the vessel wall,
Okay.
right, of the arteries.
That makes sense.
And so if you increase the
Fluid and
the permeability of the arteries, you're going to dump more water out into the tissues.